We'll go ahead and get started. Thank you all for venturing out so early in the morning for our session today on identity, biopolitics, belonging in the biopolitics of DNA and colonial modernity. This is, just so you know, the first of a two-part panel. The second half will be held in Mile High 4A after the break. And I do want to note that we have one change from the program today. We'll be having a presentation from Fatima Jackson, who's not listed on the program, at 9:15, followed by John Marks as our discussant at 9:30. In both sessions today, we'll be considering the emergence of DNA as a potent and often problematic device for producing identity, connection, and belonging. Panelists will explore the ways in which genetic and material claims to identity are constructed in colonial modernity. In this world, identities are shaped by the racial and social boundaries that exist within the settler state. And genomics becomes entangled with identity politics in ways that may reinforce, resist, or reshape colonial power structures. It is therefore important that we recognize the ways in which colonial biopolitics influences scientific practice and laboratory research, determining who gets studied, what histories are deemed relevant, which bodies count, and where they belong. In the presentations today, we'll consider the role of scientific discourse in constituting and negotiating identities in this biopolitical context. Finally, the panelists today will also explore a variety of alternative decolonizing approaches to genetic science and help us situate genetic ontologies alongside non-scientific constructions of identity and belonging. I want to go ahead and turn now to our first paper, which I'll be presenting on imposing colonial modernity on the past, narratives of identity and race in ancient DNA research. My co-author is Jennifer Raff. So over the last 25 years, it has become increasingly common for genetic research to investigate human population origins, population histories, and the biological relationships among diverse human groups. This body of research has yielded a variety of important scientific insights about our past, and it has helped to elucidate the similarities and differences that exist within and among human populations. Today, population genetic studies garner extensive media attention, as the use of DNA to trace ancestry, migration, and history has really captured the public imagination. There is also widespread public interest in this research because it is inherently biopolitical, informing our understandings of identity, group membership and belonging, and even the very nature of categories like race, tribe, and nation. In this regard, human population genomics is deeply entangled with the legacies of European colonialism, as contemporary identity politics and Western notions about the biogenetic nature of race are firmly rooted in the colonial experience. Many of the panelists today will be exploring this entanglement by examining the biopolitics of genomic research with contemporary populations. But we want to look instead at the biopolitics of ancient DNA research. Technological advances over the last decade have opened the door to a new field, human paleogenomics, in which we can extract and sequence complete genomes from ancient human remains. Ancient DNA studies therefore provide an unprecedented window into the patterns of genetic diversity as they existed in the distant past. Ancient DNA studies are also important because they help lay bare some of the enduring and problematic narratives of identity and race that persist in human population genomics today. These narratives have been shaped by the colonial encounter and the racial and social boundaries that exist in the settler state, and they have been used in the past to justify colonial oppression. We must therefore recognize that genetic claims about identity, ancestry, and history do not emerge from ancient DNA studies in isolation. Rather, they have been produced and negotiated in a particular biopolitical context. Today, we want to discuss two of the narratives that are embedded in population genetic research. We'll consider how these narratives shape the study and interpretation of ancient DNA, and show how, in doing so, they impose colonial modernity on the past. These narratives serve to naturalize certain colonial biases and assumptions, making them seem an intrinsic part of human history. We'll also show how ancient DNA studies can help to break down some of the problematic narratives that exist in human population genomics. By using ancient DNA to produce a different form of scientific knowledge, we can frame one possible path forward to help us decolonize this increasingly influential science. Okay, so the first narrative we want to discuss today is a narrative of stasis. Specifically, embedded in most ancient DNA studies, and in human population genomics research more generally, is an assumption of genetic continuity through time. In other words, it's often assumed that patterns of human genetic variation persist throughout history, linked to the same social and geographic landscapes in the past and present. It may seem somewhat odd and counterintuitive to suggest this, as few studies acknowledge making such an assumption, and many ancient DNA studies have actually uncovered evidence of genetic change through time. However, in most studies, stasis is indeed the starting assumption, the null model that is presumed to be true unless it is proven false. This model has played a critical role in many ancient DNA studies because its assumptions underlie key analyses and research conclusions. In particular, because human paleogenomics is such a new field, there's a scarcity of comparative ancient DNA data for many geographic regions. The lack of comparative data limits the inferences that can be drawn, so researchers have sought ways around this problem. By assuming genetic stasis through time, researchers can justify using contemporary samples as proxies for ancient ones, and this has allowed them to proceed with genetic comparisons that are needed to infer population histories. For example, when a 24,000-year-old genome from Malta in Siberia was sequenced in 2014, few other ancient samples from Eurasia were available for comparison. Raghavan and colleagues therefore compared the ancient Malta genome to contemporary samples from Eurasia and the Americas in order to infer the ancestry and genetic history of this ancient individual. In other words, they assumed that the contemporary samples could accurately represent the genetic diversity that existed in these regions 24,000 years ago. However, given that we know that all populations change over time, it is problematic to treat contemporary people as identical to or representative of ancient peoples, as if they had not evolved in the intervening interval. Assuming genetic constancy over time and downplaying the evolutionary forces or the impact of evolutionary forces, like genetic drift and natural selection, is very reminiscent of the ways that 18th, 19th, and early 20th century race scientists assumed long-term stasis in human population structure and the fixity of human biology. Such ideas emerged within the context of European colonial expansions to help differentiate European settlers from those they were subjugating. The persistence of similar ideas today is a legacy of colonial racial science, and this legacy has real consequences for the genetic narratives that we tell about human populations. To illustrate this, I want to return to the Malta example. By comparing this 24,000-year-old genome to contemporary samples, Raghavan and colleagues determined that the Malta individual was most genetically similar to Native Americans, which are represented by the reddish dots on this map. However, because this ancient genome also exhibited some alleles that are more common in West Eurasians, those shown in yellow, both researchers and journalists have described the Malta individual, and by extension Native Americans, as having European ancestry. In some cases, as in this article, journalists have explicitly claimed that the ancestors of Native Americans lived in Europe before migrating to Siberia and then the Americas. This narrative has been further reinforced by researchers and journalists referring to the shared genetic variants as, quote, Western Eurasian variants. Thus, the genetic narrative being told here is one in which a migration of people left Europe and carried those so-called Western Eurasian alleles to Siberia and then on to the Americas. In this narrative, European populations have been genetically static through time, exhibiting the same genetic variants 24,000 years ago as they do today. It's important to note, though, that this is not the only narrative that could be told from these genetic data. Given that the data come from a 24,000-year-old Siberian and contemporary Europeans, one could argue that the directionality of movement here should actually be reversed, and the narrative should instead be one in which present-day Europeans have Siberian ancestry. This narrative would be based on the fact that we know the shared alleles were present at Siberia 24,000 years ago, whereas the data from the Raghavan study do not actually tell us if they were found in Europe that long ago or not. However, this is not the narrative that we have been told. The narrative that has been preferentially told is one that both reflects and reinforces the persistence of colonial perspectives and racial thinking today. In particular, constructing Europeans as the ancestors in this story only makes sense if we assume stasis and a kind of genetic immutability in European populations. This assumption also produces a narrative in which the direction of movement and gene flow maintains long-standing racial ideas about the purity of white Europeans. In other words, by assuming that gene flow between Europe and Siberia must have flowed from Europeans into others, this narrative reinforces the notion that whites have remained pure while others have not. Biocolonial notions of purity and hypodescent therefore become embedded in genetic knowledge and naturalized. This narrative also fits with colonial narratives of European exploration and expansion, in which Europeans are always the colonizers. Thus, when assumptions about stasis are built into ancient DNA research, it may cause colonial perspectives and aspects of racial thinking to be inserted into our reconstructions of the human past. If we are to understand the patterns of variation that existed in the past and the genetic histories that produce those patterns, we must stop trying to impose colonial modernity on the past so that we are not simply trying to fit new discoveries into existing conceptual frameworks. Let's now turn to a second narrative that's prevalent in human population genomics and ancient DNA research. This narrative is connected to the first, but specifically conveys the idea that genetic isolation has been the norm for human populations throughout history, whereas migration and gene flow have not. In human population genetic research, we see this narrative appearing most frequently in discussions of ancestry and admixture. Ancestry can be defined in many different ways, but it's most often conceptualized as descent from discrete or semi-discrete groups that have been isolated for a significant length of time, so that they have evolved different allele frequencies and distinctive genetic profiles. Admixture, on the other hand, is formally defined as gene flow between distant and previously separated populations. In other words, admixture is the genetic exchange that occurs when people of distinct ancestries come together out of isolation and mate with one another. Population geneticists have developed a variety of statistical models to estimate the proportion of alleles in a so-called hybrid individual or population that have come from each of what's called parental populations. Integral to all of these models, though, is the notion that parental populations remain sufficiently isolated that they have stayed genetically distinct or pure prior to the start of the admixture event. Thus, in this framework, admixture and gene flow are conceptualized as aberrations in human history, exceptions to the rule. Given the contours of this narrative, it is perhaps not surprising that it emerged out of the biocolonial context in the Americas, where both scientists and non-scientists have long sought to understand the colonial encounters between people from Africa, Europe, and the Americas. Narratives of isolation and admixture therefore have deep roots in colonial biopolitics. The emphasis on population isolation and divergence, for instance, has been present in studies of human biology since the earliest days of European colonialism, when scientists sought to identify population differences in order to justify colonial hierarchies of power. Our preoccupation with quantifying admixture proportions can probably also be traced back to colonial concerns about racial purity, as can the characterization of gene flow as something unusual and out of the ordinary. When we look at the human paleogenomics literature, we see that narratives of population isolation and admixture are common there too. However, ancient DNA studies are starting to push back against these narratives by demonstrating that movement and gene flow have been ubiquitous in the past. In just the last 2 years, for example, half a dozen ancient DNA studies have been published that provide evidence for multiple ancient migrations into Europe and numerous episodes of gene flow between European and non-European peoples. These studies show that what is considered European in a genetic sense today is actually a complex amalgamation of ancestries from numerous ancient sources, brought to Europe at various points in the past. This finding, then, clearly refutes the notion that Europeans are more genetically pure than others. Thus, it is clear that aspects of the colonial worldview are still deeply entrenched in human population genomics. Many genetic narratives still discount the effects of migration and other population changes over time, and outdated conceptions of race, isolation, purity, and stasis are still embedded in our interpretations of genetic data. Because these practices reinforce and naturalize the peculiarities of colonial modernity, it is critical that we develop new and more accurate narratives. As I have shown, we can begin to do so by using ancient DNA to reconstruct the patterns of variation that actually existed in the past, and thus show where colonial misconceptions have led us astray. Furthermore, as we make more population geneticists aware of their preconceptions, and as we diversify the people involved in interpreting genetic data, we can begin to produce a different form of genetic knowledge and thus contribute to the decolonizing of human population genetics. Thank you. Our next speaker is Ruha Benjamin from Princeton University, and the title of her presentation is Informed Refusal, Salvaging the Capacity to Talk Back, or Can They Subaltern Genome Code?
Good morning. Thank you so much to Deborah Bolnick and Rick Smith for organizing this session. It's an honor to be included with such fantastic colleagues. I've updated my title a bit from the program to better reflect what I've decided to focus on, which is still having to do with power, agency, and participation in the context of genomics, but with a particular attention to the act of refusing to participate, at least in ways that comply with dominant norms and expectations. To that end, I'd like to pick up on our panel theme, Identity, Belonging, and Biopolitics of DNA and Colonial Modernity, from the vantage point of those we might call biodefectors, which is a way of thinking about those who resist biological citizenship or the rights and responsibilities that come with enhanced techniques for intervening in life itself. Biocitizenship, as many of you know, is part of a family of concepts that attempt to make sense of new forms of sociality and belonging that are growing up around technoscience. As such, biodefection helps us get at the stigma that often attaches to opting out, a conceptual corollary for us to make sense of the various forms of exclusion and resistance that also arise. So my interest in speaking back to scientific authority was first influenced by medical anthropologist Raina Rapp in her work on amniocentesis, in which she refers to those on the biomedical front lines as moral pioneers, quote, at once conscripts to technoscientific regimes of quality control and normalization, and explorers of the ethical territory that biotechnology's presence produces. For Rapp, these moral pioneers are not only those who avail themselves of biotechnologies, but also those whom she refers to as refusers and draft resistors. Both recruits and resistors alike engage in what she calls a philosophy of the limit, wherein they enact personal choices within a context of social limits beyond which they are unwilling or unable to tread. To a large extent, biocitizenship is tied to modes of consumption, such that belonging often takes shape in relation to people that make the same choices about what to consume within a pre-existing universe of possibles. For example, individuals screened for the breast cancer genetic mutation, or parents who bank a child's cord blood for future stem cell transplant. Or even more literally, people who sign up for the genetic ancestry testing company 23andMe's community portal, which is called 23andWe, through which the company connects us to people who share our genetic traits. So on the most basic level, biodefection would take the form of refusing to participate or consume the available choices in these various contexts. But for our purposes today, I'd like to imagine forms of refusal beyond the market. And for that, let me just go back one. For that, I want to draw upon anthropologists Audra Simpson and Kim Tallbear's work. Both scholars conceptualize the relationship of sovereignty to epistemology, examining forms of ethnographic resistance by informants during fieldwork and in their own refusal to represent Indigenous communities in particular ways. Simpson notes that, quote, in listening and shutting off the tape recorder, these refusals speak volumes because they tell us when to stop. Tallbear in turn explains how the concept of refusal helps frame silences as not only against the ethnographic grain, but as productive and supportive of Indigenous self-determination. This notion of productive refusal to not only negate colonial forms of knowledge production, but to create new, more just relationships between researchers, subjects, and institutions, I think should be central to our discussion. So to that end, in the longer paper, I provide a glimpse of 3 moments of refusal in the context of sickle cell disease treatment in the US, genetic ancestry testing in the UK on the border, and genomics research in South Africa, where different institutional contexts necessarily create the conditions of possibility for distinct forms of authority and resistance to take shape. But in what follows, I'll briefly discuss only the last 2, the last one in more depth, and then close with some final reflections. In 2009, the UK Border Control inaugurated the Human Provenance Pilot Project. This was an initiative dubbed to curb what was dubbed nationality swapping, wherein border agents used isotope analysis and genetic ancestry tests to see if the bodies of asylum seekers corroborated their stories. A major focus was telling Kenyans and Somalis apart, deporting the former if testing indicated that they were really Somali. Over 100 cases were vetted before the project came under intense criticism by those insisting that the underlying science should not be used in that way. What's more, for our purposes today, is that if applicants refuse the test, protocol demanded that border agents regard them with added suspicion, as indicated here, increasing their chances of deportation. In the UK case and beyond, rebuffing the biotechnical embrace of the state, to borrow Mary Jo Good's phrase, causes individuals to experience the underside or outside of biological citizenship, a form of biodefection in which refusal is always already guilty. For the second and more detailed example, we'll travel to Kimberley, South Africa, a historic mining town where diamonds and DNA share similar dynamics in terms of who tends to reap the benefit and who bears the burden. Power relations that were front and center at a genomic sovereignty workshop I participated in, which included members of the San councils from 3 countries discussing their experiences dealing with researchers from around the world who routinely seek genetic samples because the San are considered the oldest known lineage of modern humans. So top on the agenda was discussion of a 2010 paper in Nature. The investigators, who were based in the US and Australia, reported the genetic structure of the 4 individuals shown here. The study, we learned, was approved by Penn State's IRB, the University of Limpopo Ethics Committee, and the Human Research Ethics Committee of the University of New South Wales, so by all accounts, highly vetted. Readers are told that all participants consented, whether in writing or via video-recorded verbal consent, and that the collection of human DNA in Namibia was conducted under the permit of the Ministry of Health and Social Services of the Namibian government. Alongside the publication, the researchers hosted a public forum in Windhoek to present their findings. At the meeting, a member of the San community asked the researchers why they had bypassed the community councils in the consent and sampling process. In response, Australian researcher Vanessa Hayes, shown here, insisted that the team had followed established informed consent protocol to the letter, and that permission from the Namibian government sanctioned the research. End of story. But only if we view what transpired through a narrow bioethics lens. By contrast, concerned South African researchers convened a series of workshops in which council members from Namibia, Botswana, and South Africa came together to discuss issues they had both with the Nature paper and genomics research more broadly, including who has the prerogative to grant approval for such research in the first place. And while there were a number of important issues that emerged in the workshop, I'll touch on one that most closely relates to the discussion of refusal, and which grows out of the possible stigma associated with medical genomics findings. Council members wanted to know if researchers found, for example, that a certain percentage of San were affected by HIV, were they obligated to make this public? The San, like so many other oppressed groups, have a fraught relationship with biomedicine. As one council member explained, the clinics they go to gives out medicine often makes individuals feel worse. Doctors are killing us, not treating us, he said. And so discussion about whether people necessarily want this information ensued, with researchers assuring council members that people could decide what, if anything, they wish to learn from the medical genomics findings. Amidst these deliberations, and not unrelated, another important current ran through the workshop, and that's the inclusion of prayers and songs on the part of council members. Starting about midway through the first full day, one of the younger members interjected that one of the elders wanted to begin the proceeding with a prayer. Immediately afterwards, one of the researchers asked if the prayer could be translated for the rest of the group. Then, following a quick back and forth with the elder, the council member responded that just as the majority of the deliberations held in English were not translated, neither would the prayer be. But up until that point, individuals who required translation from English to Afrikaans or Setswana relied on someone willing to sit and whisper next to them. Translation, in other words, was happening in a piecemeal fashion. The refusal to translate the prayer for everyone was, in a sense, a way to insist on reciprocity and also perhaps incommensurability. Later that evening, in an informal context, the same elder who shared the prayer took the lead in singing a number of songs accompanied by a traditional string instrument that he played. In this setting, under the veranda, he welcomed translation of the lyrics, many of which were funny and mischievous, for those who were enjoying but unable to understand. The following day, then, during the session, he also opened the workshop by singing. So what began as an informal exchange in the evening became an official part of the proceedings over time. Taken together, and whether intentionally or not, the increasing centrality of prayers and songs that were not originally on the workshop agenda began to shift the balance of authority and expertise on display. This dynamic recalls a point made by Marcuse in One-Dimensional Man, where he writes, "Whether ritualized or not, art contains the rationality of negation. In its advanced positions, it's the great refusal, the protest against that which is. The modes in which man and things are made to appear, to sing and sound and speak, are modes of refuting, breaking, and recreating their factual existence." So by infusing prayers and songs into scientific proceedings, forms of expertise that underlie the facts of existence are potentially troubled. Religious, cultural, and linguistic knowledge is called forth, enacted, performed, and withheld, offering no easy transmission of cultural competency from the San to the non-San. Indeed, by incorporating prayers and songs without translation, San Council members give voice to that which is unknowable and thereby signal the limits of scientific knowledge claims. This in turn has the effect of destabilizing existing status and power relations that presume the San are there to learn from the experts. It's worth pointing out that in what they said and how they said it, the genomicists at this particular workshop sought to create a reciprocal learning environment for all involved. However, it was my impression that only after the inclusion of prayers and songs did the tenor of deliberations begin to shift and reflect the desired reciprocity. In other words, what matters is not only the intentions of those gathered, but also the structures of participation, modes of inclusion, and assumptions about what forms of knowledge and expression are valid and relevant. In conclusion, then, I don't think it's enough to simply acknowledge that refusers are justified in their distrust of medical and scientific establishments. After all, the practice of acknowledging distrust in a sympathetic vein is now commonplace among clinicians and researchers. A substantive approach to the issue, I think, requires a complete reorientation to it, away from the ethno-racial and cultural identities of problem people, to borrow from Du Bois, toward the relative trustworthiness of institutions, including those that have institutionalized bioethics. The notion of informed consent assumes that adequately conveying information should lead reasonable individuals to grant permission. How would it change the terms of engagement if subjects' refusal to participate were more fully institutionalized in the language and protocol of testing and tissue procurement, beyond the assurance that they can opt out at any time? And here I think the homograph, refuse, that is something discarded as worthless, and refuse, that is the act of declining a request, offers a discursive starting point for rethinking standard bioethics and valuing everyday forms of resistance. The purposeful conflation aims to salvage the idea of refusal and constructing more reciprocal relationships between institutions and individuals, one in which biodefection is not stigmatized, but rather stands as a necessary critique of the assumptions and excesses of forms of belonging that rest so heavily on biological claims. With that, thanks so much for your attention. I look forward to hearing your comments.
Next speaker is Rick Smith. The title of his presentation is In Cold Blood, Scientific Silence and the Constitution of an Indigenous Past.
Good morning, thanks for coming. The biological origins and early population histories of Native Americans are the primary and most visible areas of genetic research on the indigenous peoples of North America. In many cases, research into indigenous population histories involves sampling only a subset of Native individuals who trace most or all of their ancestry to the indigenous peoples present in the Americas prior to the arrival of Europeans. This approach excludes individuals who are known as admixed, those who trace their ancestry to both indigenous as well as non-indigenous populations. The goal of these practices is to include only those individuals whose DNA can be used to study the deep migratory history of Native Americans, and exclude all those whose ancestry reflects the more recent population histories of settler colonialism. As a result of this approach, much has been learned about the initial peopling of the Americas, while far less is known about the population processes after the initial colonization of the Americas, or the genetic impacts of settler colonialism resulting from disease, warfare, genocide, and forced relocations. Thus, the dominant paradigm of genetic research in the Americas centers around only certain indigenous bodies and histories to the exclusion of others, and the boundaries between scientific discourse and silence have helped shape how indigeneity comes to matter as a political subject in the colonial consciousness. But what constitutes Native Americans as a category does not pre-exist in nature, passively waiting to be described by a geneticist. Rather, this category is material discursive, emerging in and through colonial legacies, where what is knowable about Native American DNA is shaped by the ideals and expectations of Western patrimony. In turn, the conventional genetic research has often served to naturalize the social conditions of colonialisms through its characterizations of the indigenous body and the indigenous past. According to Karen Barad, scientific apparatuses constrain the material conditions of possibility and impossibility of mattering. They enact what matters and what is excluded from mattering. Throughout this paper, we follow Karen Barad's usage of matter and materialize in a biosocial sense, which indicates both the social significance and power of science to shape political subjects and the physical or corporeal aspects of scientific subjects themselves. Our goal is to evaluate the boundary-making practices entailed in genetic research with Native Americans in order to understand how colonial narratives are materialized through the indigenous bodies and how alternate decolonized narratives might come to matter. Drawing on Kim Tallbear's BioPolitics of Tribal DNA and Scott Morganson's Queer Biopolitics of Settler Colonialism, we incorporate recent theoretical insights directly into scientific practice to demonstrate how certain indigenous bodies are materialized to the exclusion of others, and how settlers negotiate their own entitlements through narratives of the disappeared native. Building on these insights, we move towards a decolonizing genetics, where colonial expectations about indigenous materiality are reoriented with regard to tribal self-determination. This approach imagines a way to proceed through the colonizing spaces of science to unsettle settler subject formations. Inspired by the insights of decolonizing thinkers like Linda Smith, we hope to provoke some thinking about the roles that knowledge production, knowledge hierarchies, and knowledge institutions can play in social transformation, in which various stakeholders reach out in collaboration to create new possibilities together. In past genetic studies, tissue samples for genetic research were collected from individuals largely on the basis of their tribal affiliation. However, in many cases, only a subset of those samples were ever analyzed for population history research, and others were omitted from genetic analysis based on their non-indigenous or multi-tribal ancestry, self-reported. This indicates that scientists have used different criteria for genetic sampling during various stages of research. Initially, they seem to use tribal affiliations and self-identifications in determining whose blood should be collected. Next, they use factors such as blood quanta and self-reported ancestry to select the fraction of samples deemed most relevant to a particular area of population research. Thus, there have been different criteria for identity and belonging at different stages of scientific research, which dynamically negotiate and renegotiate who counts as native between the syringe and the laboratory bench. This reflects a complex set of decisions on the part of genetic scientists where what constitutes indigeneity is progressively filtered by the boundary-making apparatuses of scientific research. Thus, while many individuals are socially and culturally cohered as native, only certain indigenous bodies are allowed to matter. Many of these unanalyzed blood samples have been maintained in laboratory freezer collections for decades, and the history of settler colonialism become literally deanimated in cold blood, deemed irrelevant to dominant scientific discourses on population history. We therefore undertook a genetic analysis of these samples to reveal the consequences of sampling decisions in previous scientific studies of Native American population history. We also sought to evaluate how genetic interpretations of indigeneity change when we reorient modes of knowledge productions in science using tribal ontologies and self-reported identities, rather than excluding samples that do not fit within the criteria of ancient migrations research or within traditional scientific definitions of purity. So today I present a new genetic analysis attempting to address some of these issues. And in this study, we extracted and analyzed 84 blood samples that were previously collected by other researchers from self-identified Native Americans, but which were excluded from past studies because they reported having non-Indigenous or multi-tribal ancestries. In order to contrast our results with previous studies and to determine how previous genetic research has negotiated what counts as native, we sequenced a portion of the mitochondrial DNA. This portion of the human genome is inherited solely from one's mother, and because it represents only one line of a person's ancestry, their matriline, mitochondrial DNA has been widely studied in human migrations research over the last several decades. We also analyzed differences in the length of the amylogenin gene, which can be used to detect the presence of X and Y chromosomes in each individual. And for the fraction of samples who had a Y chromosome, we assessed a short region of DNA found there that can be used to identify one's patrilineal descent. Because some tribal members are concerned that studies of genetic exchange in their communities could have potential ramifications for federal recognition status and issues of tribal sovereignty, we conducted our analysis at the regional rather than the tribal level to maintain anonymity as much as possible. We thus grouped samples into 3 major geographic regions in North America, which we'll refer to here as the Southeast, Northern Plains, and West. For each of these regions, we calculated the genetic diversity of mitochondrial DNA sequences, and to detect changes in genetic diversity resulting from the addition of previously unstudied individuals, we compiled 2 data sets shown here for each of the 3 geographic regions. In the first data set, highlighted here, contains only previously published data on presumably genetically pure individuals. The second data set, highlighted here, contains a composite of new data that we collected along with previously published data. As expected, and just pay attention to the red here, as expected, when all tribally affiliated individuals are included in genetic analysis, we detect increases in genetic diversity, most notably in the West, where our estimates of genetic diversity have increased by more than 30%. Matrilineal and patrilineal sequences from non-Indigenous sources are more highly represented in this analysis than previous studies, more fully reflecting colonial histories. We identified haplogroups, or closely related DNA sequences, and compared the frequencies of haplogroups in our study with previous research for each of the 3 geographical regions. So the haplogroups shown here, A, B, C, D, and X, are those that are commonly found among Native Americans, and they're the only ones that have been identified by previous genetic studies of populations in the United States, shown here. In contrast, our study identified numerous haplogroups from non-Indigenous populations among contemporary tribes living in the Southeast and the Northern Plains. For the paternal lineages, a similar pattern is seen. While previous studies did detect low levels of non-Indigenous haplogroups among Native American tribes, our results reveal a far greater magnitude of non-Indigenous paternal genetic lineages than has been previously observed. The frequency of non-Indigenous Y chromosome sequences among contemporary tribes is much higher than that observed among the matrilineal sequences, reflecting a marked asymmetry of male settler DNA among Native communities compared to settler females. This heightened fraction of European male ancestry among Native Americans is consistent with what we already know about settler colonial expansion in North America, which was often male-driven and likely reflects histories of both sexual violence and intermarriage. Strikingly, rates of non-Indigenous ancestry among tribes is geographically patterned across the United States. showing a distinct decline from the southeast at 21% to the west, where we didn't find non-Indigenous lineages. To our knowledge, no previous study has ever yielded this result, and this is just one example of the kinds of histories that are missing from contemporary genetic discourse. This pattern might reflect a couple of different processes. First, because more early colonies were established on native lands in the east, greater genetic diversity observed there may simply reflect a longer duration of colonial settlement and the history of interactions between Europeans and Native Americans in that area compared with the northern plains and the west. Notably, there's a remarkable absence of any colonial admixture in the west, even though these individuals studied here reported having either European or West African ancestors. We did not detect evidence of that here. This may reflect less temporal overlap between settlers and natives in the West. It could reflect shifting patterns of indigenous resistance as settlers expanded westward across the continent, or may merely reflect differences in local colonization histories. When we use the maternal and paternal sequences observed among Native Americans to identify the most likely sources of non-Indigenous ancestry, we find that non-Indigenous sequences are most likely derived from the people in Western and Central Europe, Northern and Western Africa, and the Middle East. We note here, though, that the source populations depicted here are predictions based on the geographic region where a given sequence is found at highest frequency among previously studied living populations. These predictions reflect the most likely but not definitive source populations. Nonetheless, the patterns that we see here are consistent with populations involved in European settler colonialism and the West African slave trade. When we use the criteria of tribal membership as entry points for genetic analysis, rather than limiting the analysis to a subset of individuals that facilitates deep migrations research, a very different picture of Native American genetic diversity emerges. This analysis brings histories that are commonly unspoken in science back into discourse, more fully elucidating the effects of settler colonialism. Our results demonstrate that the genetic composition of Native American communities is much more diverse than previous studies have suggested. And while Native Americans have long been characterized as showing little genetic diversity, our results in the West show that genetic diversity was previously underestimated, even among those without non-Indigenous ancestors. Further, we show that individuals who have previously been excluded on the basis of their non-Indigenous ancestry are integral to understanding recent population histories within the context of colonial modernity and must not be ignored. Indigenous histories and identities are contested spaces in science, and Native representations in genetics are entangled with the legacies of racial science and settler colonial structures of power. The dominant apparatus of genetic research in North America, which is overwhelmingly focused on Native origins and the peopling of the Americas, has largely constituted Indigenous bodies as only those which carry narrowly defined Indigenous DNA, but who has been counted as Native in scientific terms and whose identity is culturally constituted as Native do not fully align. Genetic research has too often enacted what matters in terms of deep history and has excluded histories of racism and settler colonialism from mattering, along with their genetic impacts. It has also excluded the many people whose identity, regardless of their DNA, is cohered with Indigenous people. These practices, like the centuries of Western science that precede them, too often depict Native Americans in anachronistic terms, and the conventional foci of genetic research are in need of paradigmatic realignment. They produce and authenticate certain kinds of indigeneity by privileging biological affiliations over social ones, shaping who counts as Native in terms of the deep past. Furthermore, the urgency of scientific gazes directed at biologically pure natives over those who are increasingly mixed emerges from and contributes back to the imagined extinction of Native Americans and their histories as subjects of scientific inquiry. This reifies biological notions of difference and reinvigorates false but politically expedient perceptions of the disappearing native. Thus, genetics has often served the larger political structures of colonialism where settlers imagine and enact their own identities and entitlements through collective imaginings of an Indigenous past. Our research hopefully helps to uncover the genetic effects of more recent histories of settler colonialism and attempts to destabilize Indigenous identities as biologically coherent categories. We emphasize that who counts as Native cannot be determined by a DNA analysis, and imagine a decolonial genetics that opens up the boundary-making practices that conventionally serve to bring only certain Indigenous histories and certain Indigenous bodies into scientific discourse. Our results demonstrate that some of the problems in contemporary genetics flow seamlessly out of the privileges of silence, the power to leave colonial atrocities simply unspoken, and to unsee bodies and deny them their historicity. It is a privilege of false naming and of appropriation that severs Indigenous ontologies and remakes them on techno-scientific terms. There are profound consequences if we continue to maintain scientific silence on the impact of colonial histories, and our study is an attempt to reanimate histories that are so often de-animated in genetic research, but can no longer simply be ignored. Thank you.
Our next presentation is Selling Identity, the Commercialization of Genetic Ancestry Testing and Interpreting Difference. And our speaker is Sandra Sujin Lee.
Okay, thank you to Deborah and Rick for convening this double session and for including me. What I'm going to do is talk a bit about some research on 23andMe that I've been conducting. And it builds on over 100 interviews, iterative interviews of customers who have purchased the service over the last 4 years or so. So the tremendous growth of commercial genetic databases and access to online pedigrees has fueled a rapid expansion of ancestry services and products, reflecting in a tangible way the fascination and growing appetite for genetic data on personal origins. An example is the well-known genealogy company, Ancestry.com, the world's largest online family history resource, which reports having approximately 2 million paying subscribers that have created over 41 million family trees containing approximately 4 billion profiles. In 2012, the company was acquired by a private equity group for $1.6 billion. Outside of the purview of the FDA and other federal agencies, genetic ancestry testing has been free to grow unfettered by the regulatory challenges that have recently stymied genetic testing in the health arena. So this paper examines how genetic ancestry testing customers interpret ancestry, race, and social identity in the context of human genetic variation data, and how genetic information is imbued with meaning and selectively incorporated into a sense of self. In suggesting that customers play with this data is not to undermine the enduring allure and power of genetics as a precision science, nor minimize the weighty implications of genetic test results, but to note the context in which customers make sense of complex information they receive, and to interrogate that framing, the framing of the service as an experience of self-discovery. So transcending temporal and geographic boundaries, DNA ancestry testing imbues personal history with a materiality that gives individuals permission to imagine themselves as having a corporeal connection to unknown peoples and unknown times. Accompanying this new era of consumer genomics is the proliferation of initiatives to incorporate genetic ancestry testing as a vehicle for science education and genetic literacy. This paper considers the implications of using genetic ancestry testing as a tool to promote scientific literacy and the teaching moments that are to be gleaned by students in defining who they are. So most of you probably know 23andMe. It's a genetic sequencing company in the heart of Silicon Valley. It made headline news shortly before Thanksgiving in the fall of 2013 when it was ordered by the FDA to discontinue marketing of its saliva kit and personal genome service. The PGS came into the market in 2007, and over the ensuing 6-year period, the company grew to offer over 200 genetic test results that included risks for disease, including Alzheimer's disease and breast cancer, testing for carrier status and behavioral traits ranging from sprinting ability to nonverbal IQ, as well as genetic ancestry, Y chromosome, mitochondrial DNA, and autosomal testing. Envisioned as first and foremost a company concerned with health ancestry information, it was initially considered that ancestry information would be only a secondary focus of the company. The test kits were sold separately, with health information seekers predicted to be its major market. However, upon realizing that many more customers purchased the Ancestry information, 23andMe quickly pivoted and started to bundle their Ancestry information with health-related genetic testing into one product. Pitched to investors as much more than a fee-for-service business from its inception, 23andMe has maintained a biobank that includes DNA samples from their customers upon their consent. A consistent feature of the company is a biobank and the 23andMe program that aim to develop a robust DNA resource for research. In December 2012, the company announced its, quote, 1 million strong campaign to enroll 1 million customers into the company biobank, which it achieved earlier this year. Ann Wojcicki, CEO of 23andMe, predicted that a, quote, genetic data resource of this magnitude has enormous potential to address unanswered questions related to the contribution of genes, the environment, and your health. Understanding these factors and their interactions could lead to major improvements in diagnostics, preventive medicine, and therapeutics, end quote. So in 2013, the FDA publicly claimed that 23andMe's PGS was a medical device that had not received pre-market approval. Departing from years of discretionary enforcement authority, the FDA ordered 23andMe to shut down the return of health-related genetic risk information to its customers. Although access to the genetic testing results, this type of test results were not available to new customers, kits were continued to be available for purchase of the genetic ancestry information as well as the raw genetic data. So what I'm going to do is to share with you some of the excerpts from interviews that I've conducted with customers to give you a sense of the ways in which these individuals are incorporating the genetic information that they're getting from 23andMe. And I'll start with Stephen. I'm going to go to this one. Stephen, a 54-year-old self-proclaimed genealogist who works in human resources for a food supply company in Colorado. He was motivated to purchase his 23andMe kit to augment his considerable efforts to fill in gaps in his genealogy. So in addition to 23andMe, he had purchased genetic ancestry testing from Family Tree DNA and National Geographic. When we first met for our first interview, he was excited to report a novel finding that he had discovered from his 23andMe report, and he says, okay, so I know my ancestry is primarily European. I thought I'm probably 100% European, and I had heard that there were rumors in the background of distant relatives living in Germany. I was curious because one of my family's last names was Kaufman, so I asked around at a family get-together, and I said, well, is anyone Jewish in the family? And my maternal grandfather said, oh, yeah, probably. Well, I thought that same side was going to be providing some more interesting exotic relatives that I wasn't aware of. At any rate, I was curious to do some digging, but I still thought, I'm 100% European. Well, to my utter surprise, the ancestry painting came back from 23andMe, and it showed some Asian and some African. So then I started wondering, well, where is that? I don't see it. I think that the Asian can translate into Indian, which has been rumored in my family anyway. There were always talking about the possibility because we bruise very easily. I'm sorry, did you say bruise? Yes, my cousins and I have always bruised to the point where doctors would pull my aunts aside, my mother aside, and say, is there any child abuse going on here? No, they're just kids, and that's what happens, we get injury, we bruise. Well, the rumor in the family has always been, oh, it's Indian blood, gypsy blood, something like that. It had never really been substantiated until, well, that's what I was thinking when I got my results back from 23andMe, but I was always questioning my maternal grandmother. So this was my father's mother, because she does not look English or, anything Northern European like the rest of the family. And so when the results started coming back that yes, there is some Asian and some African, well, I said, oh my gosh, maybe there is actually some American Indian, but I couldn't figure out the African. So like a jigsaw puzzle, Stephen tried to fit pieces into a tapestry of his identity that retains some recognizable portrait of himself and his family. Rather than rejecting or discounting seemingly incongruent genetic results, he laces memories and physical details to validate genetics into a cohesive family story. Understanding how genetic ancestry testing and claims of population differences requires what Ahmad Macharak calls for in her analysis of the fluidity of race and the way in which it is practiced, not to be confused with the multiple meanings of race that collide in the aggregation of genetic databases and references created in the service of biomedical research and the classificatory schemes used to characterize difference, but in the flexible use of race by individuals who consume information on human differences, and the determinations about their geographical origin and group affiliation that are derived from these resources. Macharak charges her readers to take up the challenging questions of how to talk about materiality and the factness of race without embracing a naive version of the biological, and what, in the end, is the factness of race, she asks. So during my interview with 23andMe customer Stephanie, a 42-year-old lawyer of a small startup company in San Francisco, I asked her, as I asked all my interviewees, what was the best outcome for her with her experience with genetic testing. And instead of relaying her own experience, she chose to tell me about her friend Michelle, to whom she had recommended the product. Michelle was a longtime friend of Stephanie's, who was a venture capitalist also based in San Francisco. Stephanie called her typical of most of her close friends, someone who worked hard, 100-hour weeks with very little downtime, and who played hard. Stephanie was with Michelle at the time she accessed her 23andMe results online. And she says, it was sort of fun for her because as she clicked through on that particular view, she saw that she is in Africa very clearly. It was kind of fun because she doesn't know anything back to the next generation, and she very clearly maps to a particular North African tribe. For her, it was the sort of kind of thing that without access to the product, she never would have really known. Do you know what I'm saying? She looks like me, except with curly, maybe she looks kind of French or something. So Stephanie said that the genetic ancestry test results were interesting on their own, but it was understanding the results in the context of some symptoms that Michelle had been exhibiting over the preceding 6 months that Stephanie said Michelle fully appreciated her 23andMe testing. Michelle noticed a rash on her legs and went to UCSF, and her dermatologist said, I don't know what it is, but you may have an allergy to chlorine. You've been swimming more, so that makes sense to her. But a few weeks after, she noticed some ringing in her ears and went to an audiologist, and he said, gosh, you did have an infection a while back, didn't you? I mean, it could have been that there's sort of damage that came from that. It may have just taken some time to resolve. Let's watch because we don't see anything. And then she had some blurred vision and went to another doctor who said, you're getting older, your prescription may have changed, and you've really got to work on correcting that. Anyway, she wasn't that worked up about things, but it was these different components of symptoms, if you will. Anyway, nobody put it all together, and she didn't put it all together. Now, at this point, Stephanie leans over and lowers her voice, and she says, well, it turns out that she has lupus, and it's a disease that's very common in African-American women. But even she, as genetically an African-American woman, very clearly didn't think of herself even that way looking in the mirror. And none of her physician friends had. Whereas in retrospect, imagine had we known. So she looked at 23andMe risk ratios, and she had an extremely elevated risk of lupus, end quote. To Stephanie, genetic ancestry information is what was most valuable to her friend Michelle in finding the answer to her medical symptoms, rather than the genetic test result itself on lupus. Michelle's hidden, quote unquote, hidden racial background that was illuminated by genetic testing was in Stephanie's mind the key to understanding the relative risk for disease, and was it was for her the key to diagnosing Michelle. She explained, it was a sort of pulling forward of phenotypic data that should have been observable. So if an African-American woman were to come into a doctor's office with these kinds of symptoms, a typical internal medicine doctor would probably put these pieces together saying, you know, to somebody of this genetic background with this symptomology, we see lupus sometimes. But because it was so unusual in a Caucasian woman, the pieces didn't come together, unquote. So contrary to the mantra among many scientists that greater understanding of genetic ancestry will render the racial categories as obsolete, Stephanie sees genetic sequencing as a tool for pulling to the surface that which would be otherwise invisible. The genotype reveals innate racial identity. This interpretive act is what Anne Morning, in her study of race in biological textbooks, notes as seductive about genetic race, as a perfect kind of race for a multiracial American, because it removes the requirement that racial membership be visible or obvious to the common person, that it look like anything in particular at all. So genetic ancestry is no longer only for the fervent genealogical hobbyists willing and able to purchase testing kits. Rather, it has been increasingly embraced as a tool of choice in increasing genetic literacy in the broader public. And I'm just going to allude to one program that was recently announced. So a new program based on Henry Louis Gates' popular PBS documentary, Finding Your Roots, recently received 2 grants, including $355,000 from the Robert Wood Johnson Foundation and another $300,000 or so from the National Science Foundation for a college program. The Finding Your Roots middle school project will focus on evolutionary biology and health lessons, as well as provides hands-on activities for middle school students attending summer camps at Penn State. And there are a number of different programs that have been introduced. Explaining the rising need in the U.S. to increase diversity within its workforce, in particular science, technology, engineering, and mathematics, the programs cite the disparity of African American and Hispanic professionals that make up 12 to 16% of the STEM workforce, and the declining percent of underrepresented students pursuing STEM degrees. And it has become a major justification for using genetic ancestry for public literacy. And here Gates explains, I conceived of this project after the first session of our PBS series aired in 2005, as I realized the impact that tracing one's genealogy can have on the self-esteem of young people of color. As every amateur genealogist knows, reconstructing one's ancestry is just another way of learning about yourself. Far too many of our children have had their passion for learning crushed in poor learning environments, both at home and in our schools. Learning the art and science of ancestry tracing through DNA and archival records can help to regenerate that passion for learning because it is so personal, and besides, it is fun, end quote. So Gates asserts genetic ancestry is not only a vehicle for scientific literacy and an intervention on disparities on education, but inextricable from the same rhetoric used by 23andMe, in which genetics becomes a portal of self-discovery and fun. This framing reduces the complex historical basis for educational disparities and processes of racialization that are embedded in the genomic technologies themselves. The history of sampling of populations and the creation of genetic references used to return results to the public are occluded from view as biological factness. So in conclusion, I want us to just think about Michael Flowers and Deborah Heath's prescient article in 1993, in which they describe consensus inscription when talking about the Human Genome Project. The first focuses on how scientific knowledge is developed through a system of literary inscription, leading to the creation of material traces of scientific inquiry by laboratory apparatus that characterizes and displays the presence of DNA in graphic form. The enticing figures that animate the genetic ancestry results from 23andMe and the reveal that culminates each episode of Finding Your Roots is an example of the process of material inscription of identity. The second element, consensus, or the agreement among social actors on the questions, are both constitutive of the dimensions of knowledge, yet whose origins and function in narrowing the possibility of scientific answers are rendered invisible. As Flowers and Heath wrote about the process of fact construction, once a document is hypothesized as fact, evidence of earlier inscriptions of its social political history fades. Similarly, the aggregation of DNA samples and the political historical paths of sorting difference and the basis of creating terms such as admixture fall away as genetic lineage becomes the facts of human identity and life history. Despite arguments that genetic ancestry will teach genetics to the public, there is a lack of space to inspect genetic data and the process by which it is reconstituted in ancestry information. And finally, this third element of conscription, through a beckoning of the public to engage with an offering of their saliva sample and to support the goals and existing arrangements of institutional power that integrates this into genetics. Genetic ancestry is consumed to not only tell us where we come from, who we are, and the life we are likely to live, but it now, apparently, provides solutions to inequity and disparity in education, all the while keeping it fun. So I'll end there.
Our next presentation is Rehabilitating Genomics, Indigenous Governance of Genomic Research in Australia by Emma Cavall.
Thank you, Deborah. It's wonderful to be with this group of colleagues, and how amazing that your BioAnthro lab exists that produced this incredible social science scholarship. So in this talk today, I want to do three things. First, I'll tell you about the establishment of the National Centre for Indigenous Genomics, the first purpose-built Indigenous-governed biobank in the world. Second, I'll show you a 4-minute animation I produced to explain the idea of the Centre to Indigenous community members and other audiences. Third, I will tell you the top 4 things about this project that keep me up at night. Before I start, though, the talk will focus on the practical work I'm involved with, as I think this is critical to bring to the discussion today. But I want to tell you I'm also, alongside my practical engagements, I also publish on the anthropology of genomics and indigeneity. And here are some recent papers. You can have a look if you're interested. And I'm pursuing this work at the moment in a book project entitled Haunting Biology, Science and Indigeneity in Australia. I'm an MD, PhD with nearly 20 years of experience working with Indigenous Australians as an activist, doctor, public health researcher and cultural anthropologist in roughly chronological order. For the last 8 years, I've worked on the ethical, cultural and political implications of genetics for Indigenous Australians. When I started on this project, there was very little to study. Almost no genetic research had occurred in the country. Genetics was a stigmatised science associated with the paradigms of racial classification and government control that Indigenous Australians had fought against from the late 1960s. It was very much in a state of refusal in relation to genomic science. By the time the Human Genome Diversity Project reached Australia in the mid-1990s, we'd already developed a separate ethical process for Indigenous research, and this is the latest reiteration of that from 2003, developed by the National Health and Medical Research Council. There was no way that Indigenous people were going to participate in the Vampire Project, and they did not. So when you see papers that map global genetic data, and here's just one example randomly, Australia will usually be empty. When I began working on genomics, I was sceptical about its potential benefits, but I was convinced that Indigenous people needed to be thinking through its implications. In 2010, together with the Lowicha Institute, Australia's national Indigenous health research organisation, I organised a national roundtable on the use of Indigenous genetics in health research, which brought together Indigenous community members and researchers, ethics committee members, genetic researchers and genetic literacy experts for the first national discussion on how genetics could be used in Indigenous health in a way that offered potential benefits while avoiding harms, largely through strong Indigenous oversight. This started a national conversation in Indigenous health circles, which I think has led to more projects using genomic methods today. That same year, I first made contact with the Australian National University, the ANU, who I had heard had a large collection of Indigenous blood samples from the 1960s and 70s. The collection of about 7,000 samples from over 50 communities was assembled by Bob Kirk, a British-born human biologist, and this is Bob Kirk in the field. He developed broad networks for collecting samples that were analysed in his state-of-the-art starch gel electrophoresis lab at ANU. Some people who donated samples might have been vaguely aware the purpose of sampling was to look at human variation and how different groups were related to each other. Many other samples were collected for clinical purposes or for other research projects, and a portion of the samples sent to the ANU. Samples were used to discover new blood protein types and map their prevalence across Australia and the region to make inferences about human migration. In the 1980s, DNA was extracted from a few of the samples and used for research on microevolution and population genetics. By the late 1990s, both the Diversity Project and the repatriation of Indigenous human remains from museums made scientists at the ANU worry that the blood samples they held shouldn't be used any longer without asking Indigenous people. The collection transformed from a scientific asset into an ethical liability and was closed to scientific study. When I made a cold call to the Professor of Genetics, Simon E. Steele, in late 2010 to ask about the collection, he had been waiting for my call for over a decade. Over the last 5 years, I've been working with the ANU to bring the collection into the ethico-political regime of the present. This involved forming an Indigenous consultative committee of eminent Indigenous Australians to advise the university on what they should do. I was engaged as a consultant to write a long report on the history of the collection, potential ethical and cultural issues, and options for what could be done with it. The committee decided that the collection should be retained and set up as an Indigenous governed centre. Early last year, the National Centre for Indigenous Genomics was launched with an Indigenous Majority Governance Board chaired by Mick Gooda, the Aboriginal and Torres Strait Islander Social Justice Commissioner at the Human Rights Commission. The board also includes Indigenous scientists, lawyers and health researchers. And just in this image from the launch, Mick Gooda is on the right-hand side, Mick Dodson, another board member, is on the left, Misty Jenkins, another board member, is next to him, and then the other two women are Bob Kirk's two daughters, who are not on the board but were at the launch. The purpose of the centre is to, quote, establish a national resource under Indigenous governance for appropriate and respectful genetic and genomic research that will benefit Indigenous Australians. These are, we have a list of current priorities, but the major one is to consult with people who donated samples. There it is, there's our list of current priorities. Our major goal is to consult with people who donated samples, their descendants and communities of origin, and request consent to include their sample in the centre. We are still near the beginning of this process. Not one sample has been consented or refused. But we felt it was important to create an accessible account of how we saw the process unfolding. And by we, I mean centre staff, board members, and other Indigenous individuals and organisations that advise us. So what I want to do now is show you this 4-minute video.
Imagine your veins are like rivers and your blood is like their waters, with lots of stories to tell. They actually let us know where we come from and explain to us why we get sick. These stories are already in the blood in our bodies and are written in language that doctors and scientists can understand. These stories are called DNA. And the stories come from both our mothers and fathers. They got the DNA stories from their mothers and fathers, who got it from their mothers and fathers. Because this has been going on for thousands of years, DNA stories can tell the story of a very, very big family. People share some of their stories with other people from the same area, even if they are not from the same family or clan. So when we look into the story of the DNA for a person, we also see a little part of the DNA story of their ancestors from thousands of years ago. Understanding the DNA stories can help scientists to understand more about why some families and communities have lots of people with diseases like diabetes and kidney problems, and other families have only a few. It can also help us understand why medicines work well for some people, but for other people they don't work or can even make them sick. Long time ago, small bits of blood called samples were taken from Aboriginal people in communities all over Australia. Right now, the samples are being kept at the Australian National University in Canberra. A mob called the National Centre for Indigenous Genomics, NCIG, are looking after them. This mob have a board with mostly Aboriginal people. They know it's important for us to know about our blood and our stories. They are not really sure how much those old people knew about giving their blood samples. So they think that their families should be able to decide now what to do with them. If you and your family decide that the sample should stay in the collection, the NCIG mob would get the DNA story out of the sample and read the story of the blood onto computers. The DNA stories from each sample will be put together with the stories from all around Australia to make a library. The stories in this library computer will have no names, only the DNA story and the community where the sample came from. Sometimes researchers, like doctors, who are looking to find out more about diseases might ask to look at some of the DNA stories. They will have to ask the NCIG mob first. If the NCIG mob think that their project could help Aboriginal people, they will let them read the story, only for that project. The NCIG mob are being very careful to make sure that Aboriginal people are in charge of decisions about who can read the stories and what they can read them for. You'll be able to see what the sample is being used for on an app on your mobile phone or computer, so you can say no anytime you want to using that app. This is a very new thing for everyone, but the NCIG mob want to make sure that the DNA stories can be used to help Aboriginal families and all Aboriginal people. Keeping these stories might be very important for all Aboriginal people. What do you mob think about that?
I'm aware that this animation raises as many or more questions as it answers, and I hope to address some of your questions in this session and afterwards, but in the few minutes I have left, I want to share the 4 questions that most trouble me personally. 1. Have I become a gene pusher? When the scientist I collaborate with is asked about the promissory benefits of genomics for Indigenous people, what are they? He responds with a remarkable tale of Inuit diabetes. I find myself telling it too. A study of Inuit in Greenland found a significant minority of the population have a gene variant associated with a 10-fold risk of diabetes. And when those people have diabetes, it's hard to control. So they may have been told they are non-compliant with diet and medication, when in fact they have a different variant of the disease that probably needs a different treatment. In this tale, gene variants become anti-racist, a way to shift blame to scientists and clinicians who inadequately recognise a different diabetes syndrome. I still believe the social determinants of health are more important to address than genomics, reflecting my background in Aboriginal health. But I don't buy the argument that money spent on genomics takes money away from more worthy things that you often hear. But I'm also aware that this may be related to the fact that very little money is spent on genomics in Australia. I'm persuaded by the argument that Indigenous people deserve to be included in any benefits of genomic science, although some social scientists are convinced that rebiologisation of race inhibits group-based solidarity. I don't think that's clear yet to us. So, am I a gene pusher? Or have I worked through my social science genetic essentialism baggage to objectively see the potential benefits? 2, am I creating an ancestry test for Aboriginality? Anyone can send some spit to a genome company and find out their Native American ancestry fraction. And this is one for a prominent blogger. You can't do this for Indigenous Australian ancestry, although I know of a few private companies that have tried or are trying to make it possible. A side effect of the centre is creating the data that could be used to create an ancestry test. The governance board is wary of this and would not allow access to the data for this purpose. But since we have begun consulting over the last 18 months, some community organisations have expressed interest in using the data in two ways that could create de facto ancestry testing. One, for provenancing unprovenanced human remains, and two, for establishing family connections for descendants of what we call the stolen generations, children of mixed ancestry taken from their homes and institutionalised. Ancestry testing may be an unavoidable price for inclusion in genomic research. Creating a definitive genomic ancestry test within an Indigenous governed structure offers many advantages over leaving it to the commercial market. But is it possible to avoid genetic ancestry testing altogether? And would this be a better outcome for Indigenous people? 3, is it justifiable to add to the sum total of worries of Indigenous worlds? Earlier this year, 2,693 blood samples collected in 1967 and stored in various labs in the US were returned to the Yanomami people of Brazil. As part of the campaign to have them returned, the Yanomami made a video outlining their case. It explains their view that the souls of the dead will not rest if a part of their person remains in this world. And as an aside, this is not a view that's been expressed in Australia so far. Watching the video a few years ago, one quote jumped out at me. An unnamed Yanomami elder says, "If I didn't know, I wouldn't mind." He goes on, "But this whole blood business keeps hanging around in our minds." Leaving aside questions of the adequacy of translation, the sentiment in this quote worries me. Would it be better if old blood samples were destroyed without calling on their owners or donors to speak for them? In our efforts to address the problem of frozen blood samples, in our effort to address the problems these samples pose to institutions, are we unjustifiably worrying Indigenous people who already bear vastly more than their fair share of emotional burdens? Am I participating in heritage creep, where the circle of tangible and intangible things we are called on to care about ever widens? As my time is just about over, I'll just ask my remaining question and leave it hanging. Is it okay to take these risks because the centre is Indigenous governed? Or put another way, if this is the molecularisation of identity, the reinscription of race, strategic biological essentialism, or however you want to put it, is that okay? I'm far from the first to be asking these questions, and I'm lucky to have so many great colleagues to think with, including everyone on this panel today. Thanks.
We have a slight change from the program now. Fatima Jackson didn't end up on the program, but had prepared an abstract and a paper for this session, and unfortunately she can't be here today, so she asked me to present her paper for her. And so this is a presentation by Fatima Jackson and Christopher Cross exploring the effects on African American identity of ancestral genetic composition. So I'm speaking with her voice now. We appreciate this opportunity to present our early findings on the effects of ancestral genetic composition on African American identity. Genetic analyses have been invaluable in elaborating and often clarifying a people's population history. Studies of the genetics of African diasporic populations are in their infancy, largely because of the lack of interest by majority population researchers, limited historical knowledge of the origins and dispersions of New World Africans, and a paucity of meaningful collaborative teams of researchers exploring population substructuring in the African diasporas. Interest in the genetic history of African diaspora populations was spurred by African American complaints of exclusion in both the Human Genome Diversity Project and the Human Genome Project. These complaints later crystallized around the Manifesto on Genetic Studies among African Americans, issued in 1994. Soon thereafter, we began to see more federal funding available to study African American health disparities, primarily through the emergence of the NIMHD, the National Institute for the Study of Minority Health Disparities, including the contributions of African American genetic and later genomic studies to these differences in health outcomes. More resources have subsequently emerged to detect so-called ancestry informative markers, or AIMs, and these studies have presented new challenges to the issue of identity. The question of whether we can meaningfully set parameters on African American biodiversity took on a genetic focus. With release of the genetic results of a subsample of the New York African burial ground in 2004, it became clear that some level of ancestral verification could be achieved through the study of mitochondrial DNA in African American ancestral populations. As our technical abilities increased, detection of Y chromosome augmented the mitochondrial DNA studies, and the database grew of genetic polymorphisms frequently found in African Americans. The historical context of the quest for ancestral identity among African Americans is best exemplified by this quote from Frederick Douglass in 1845. The peculiarities of American slavery systematically destroyed the foundations for a robust knowledge of ancestry. Without benefit of original names, language, tribe, and clan affiliations, African geographical regions of origin, and the intentional destruction of families left major wounds in the psyche of many African Americans. Thus, ancestral reconstruction has historically been considered a high priority with African American ethnic groups. The search for relatives separated by enslavement was usually the first task assigned upon emancipation. Often, full reunification of families was impossible. In the United States, identity is disproportionately linked to one's phenotype, and identity can be associated with levels of self-confidence, performance, and overall positive life outcomes and behaviors. In this small study, informed consent was obtained from 5 men and 6 women, all self-identified African Americans, students at Howard University, biology majors, and were all aged between 18 and 23 years. Each participant was given a pre-survey questionnaire that asked about who they thought they were descended from. Saliva samples were then taken from each and submitted to 23andMe for genomic evaluation. Students received their confidential results directly from 23andMe and then shared these with the authors. After receiving and reviewing the results of their genomic evaluations, participants completed a post-survey questionnaire. The gist of these post-survey questions centered on the interpretation of their genetic results and any changes in their perceptions of ancestry. Later, in a group setting, the pooled results of the genomic evaluations were discussed, and the historical context for most of the findings were presented. Students fell into 3 main categories of response. There were a few students whose genetic identity matched with the expectations from their family stories and their phenotype. For example, 2 students who considered their biological lineage to be 100% African in origin were confirmed by the genomic analyses of each chromosome. In these cases, one student was identified by 23andMe results to be genomically 98% West and Central African, while the second student was identified as 89% West African. Students with more distanced family structures, i.e., students who knew little of their family history or who had not spent extensive time among their immediate family members, were able to use the genomic evaluations to either confirm or deny some of the verbal accounts of their history. For example, a student with mixed West African or South Asian heritage who had been an orphan as a child was able to use the genomic data to assess the story she had been told about her mother and father by her grandmother. In most cases, the genomic results generated from the ancestry testing matched student expectations. When something unexpected was observed in their reports, they sought to quickly reconcile the unanticipated finding. For example, a student with an unexpected East Asian component of 11% of his genome reinterpreted this as actually evidence of Native American ancestry. He felt the company was in error in assigning the variation to East Asia. A review of our study results and a review of the literature suggests that regional variation exists in the proportions of Africans brought from specific areas of continental Africa that may be reflected in the probabilities of findings, particular patterns of the students we studied. Increasing our sample size in future studies and making inquiries as to the U.S. origins of the students will assist in addressing this issue. Initially, we hypothesized that all of the students would exhibit African mitochondrial variants, but we now think that there is some low level of cryptic non-African mitochondrial DNA evident among African Americans that may be due to historic and/or cultural events. Without detailed family histories on each student, it is difficult to determine when these non-African genomic variants entered the lineages of these African American students. African Y chromosome variants are reduced in the Americas because of the gender-skewed nature of American slavery and the historical introduction of European Y chromosome variants into the African gene pool. One student did exhibit an old African Y chromosome variant, which will be discussed later. It is clear from our preliminary findings that genetic testing has an impact on the identity of African American college students. 80% of the students reported some change in the way that they viewed their heritage post-testing. Ancestry components were frequently reinterpreted to increase the affirmation of specific aspects of identity. For example, a student with the Y chromosome I haplotype, a variant commonly found in Scandinavia, Germany, and Scotland, was reinterpreted by the student as actually coming from, quote, brown people, since the coalescence time for this variant was before the emergence of some of the genes responsible for decreased skin pigmentation in Europe. Another male student, who was himself very light complexioned, was visibly empowered when he received the report that his Y chromosome was a very old African haplotype A, near the root of the Y chromosome tree. A third student discovered that he had a Y chromosome haplotype that was identical to that found in Ramses III from Egypt. This became a source of pride for him. Other ancestry components were selectively ignored or de-emphasized by the students. For example, in large measure, genomic evidence of European admixture in excess of 40% of the total genome was de-emphasized. Genomic evidence of any Neanderthal admixture was dismissed. Genomic evidence of South Asian and East Asian ancestry was discounted. Most importantly, genomic evidence of adverse health predispositions did not seem to trigger concerns on the part of the students. The results did, however, generate increased interest in the historical context of individual ancestral genomic evaluations. Students were interested in why and when a particular genetic variant was present in their genomes. As frequently, they were interested in what was revealed by the test. In our general class discussion of human evolutionary genetics, the information on coalescence times for particular haplotypes was only partially internalized by the students. Our impression is that while the students, all of whom were biology students, had a better grasp of the time depth than the general public on some of this, the concept was still difficult. Fortunately, 23andMe does not issue ethnic identity certificates, as do some of the commercial ancestry testing companies, such as African Ancestry, and the students had had previous exposure to the reality that multiple African ethnic groups may share particular polymorphisms. From this small study, we were able to draw a few important recommendations. First, it is clear that the best genetic ancestry testing results are those that are most comprehensive. Isolated uniparental markers, such as mitochondrial DNA or Y chromosome markers, are not as informative and are more disruptive to the student's previous identities when used as sole indicators of ancestral lineage. Second, limiting the analyses to uniparental markers masks the tremendous ancestral heterogeneity observed in the genomes of our participants and actually does a disservice. Third, students were more receptive to the genetic ancestry results presented by 23andMe when they had less previous information about their immediate biological lineage. This seems counterintuitive, but may reflect the limited niche available for ancestry in our consciousness. Finally, genetic ancestry results that contracted family traditions tended to be discarded by the students as errors and were not discussed extensively. We believe that it takes time to internalize information about ancestry, particularly when it is complex. Genetic ancestry is no different in this respect. It would be valuable to revisit the participants a year later to see which aspects of their 23andMe reports have been accepted and possibly acted upon. Thank you very much for your attention to this presentation. We welcome your comments and questions. Okay, so we'll now invite up Jonathan Marks, who is our discussant for this section.
I've always wanted to do that. All right, well, I'm very grateful to be invited into this session and to engage with some of the work here of very interesting younger scholars, although I realize that sort of compared to me, everyone now is sort of a younger scholar. What I'd like to do is to free associate and relate some of the things that have been discussed today with things that I remember and experienced a couple of decades ago in the field of human population genetics and genomics. So there are two general themes that sort of strike me that have come up from the presentations today. One of which is how reactive contemporary work in this area is to the work that was being done in the 1990s by the Human Genome Diversity Project led by Cavalli Sforza. And secondly, how what goes on today in the field of genomics is science, but it's a different kind of science than the genomics or genetics of 20 years ago, because genomics today takes place in a neoliberal capitalist context. And sometimes, quite honestly, it's difficult to tell what the truth content, what the truth value of science that is done for profit really is. I'm stuttering, sorry. But this sort of gets to some of the points that were made by Emma Koval a few minutes ago. When you talk about genomics and the reason to do genomics, why participate? Because it's fun, is a very strange motivation for science as far as I'm concerned. After all, ancestry testing, I can't even read my handwriting here. Oh, there we go. If you read the fine print in the ancestry tests, you know, the fine print says it's not, the results you get are not legally meaningful. That's why it's recreational, because it's fun, and that's why you should do it. You can't use these results to make any kind of a legal argument. And that's also, I think that gets to some of the ideas that were expressed in Fatima Jackson's paper about how the students that took these tests made their own meaning of the results that they got. The meaning that came was not self-evident, it was not authoritative, but rather it was negotiated, it was constructed, sometimes it was ignored, and it was often reinterpreted. Let's see. I have some really interesting things to tell you, if I can find them. Okay, I'm a little scattered. Just bear with me. Everything will be fine. Let's see. Note number 3. Oh, yeah. Any of you remember the 1991 paper in PNAS by Bocock et al. That showed that Europeans are a 65-35 genetic mixture of Asians and Africans, in which the sample size by which they determine this, and it's published in PNAS, a very reputable journal. And the sample that they used to determine that Europeans were this sort of martini, or this genetic martini, was the Africans were 95 pygmy samples from Central Africa. The Asians were 110 people from Chinatown, and the European sample were the guys in the lab. Okay, and they used this and generated these sort of broad conclusions about broad populations. And it's wonderful to see. I mean, it's weird to think that this got published even in 1991, but it also is very nice to be able to see how the field has evolved since stuff like that was being claimed and said. On the theme of biodefection that Rua Benjamin brought up, you know, I'm thinking of the eugenics movement. That's older than me. I wasn't there in the eugenics movement. But back in the 1920s, the eugenics movement was very much a science oriented and science self-starting project. If you were against the eugenics movement, you were perceived as anti-science, anti-evolution. And what's sort of interesting about it is to look retrospectively at the people who were against the eugenics movement, who opted out of it and who rejected it in the 1920s. By the 1930s, all the geneticists were saying, okay, this is silly. But in the 1920s, the people who were against the eugenics movement were the anthropologists, Franz Boas saying, you know, these are, civilization is not a genetic property and you're reducing historical processes to genetics falsely. You had civil libertarians like Clarence Darrow, who was against the idea of the state controlling reproduction. You had Catholics who were against it because they were against any human intervention in a divine process of reproduction. You had, and then you had political conservatives because, after all, the eugenics movement was a progressive scientific movement designed to improve society scientifically. And it was a very weird and broad coalition. But if you were in there at all, you were perceived as anti-science. And you know, the question that Ruha Benjamin's paper raises for me is, how do you, in the contemporary world, how do you opt out of genomic research without being tarred by the same label as the anti-vaccine people and, you know, sort of general anti-science stand out there? It's a difficult line to walk, I think, in the modern world. So I'll be very interested to hear how that pans out. Let's see what else. Oh, back in about 1993, this is a hilarious story, again, and I can tell you're all sort of laughing at this. But when I first began to critique the Human Genome Diversity Project shortly after its announcement in 91, and by the way, one of the cool things is if you live long enough, like apparently I have, you get to actually write the entry on the Human Genome Diversity Project for the Encyclopedia of Bioethics. And that really pleased me because, by God, 20 years ago, I was getting so much shit for saying anything critical about the Human Genome Diversity Project. But a very senior cultural anthropologist at Yale buttonholed me in the hallway about 1992, 1993, and he said, why are you making all this fuss about, you know, informed consent and stuff like that? Back in 1962, when I went to the Kapauku, I collected 100 samples of blood for John Buchner-Janisch, who was analyzing it, and I didn't have to worry about informed consent. I just, you know, got their samples and brought them back, and everything was cool. Why are you making such a big deal of it? And I said, well, let me ask you something. Did they have, like, taboos about their blood? He said, you taboos? You bet they had taboos. It was not so easy to get their blood. And I said, well, then how did you get them to violate their taboos and give you their blood? And he said, well, I went there with samples of penicillin. And if they gave me the blood, I gave them penicillin. And if they didn't give me blood, they didn't get penicillin. And I said, so what you're saying is that if they participated in this research, they could live? And if they didn't participate, there was a health risk involved? I said, yeah, what's the problem with that? I said, well, you know, that would probably not pass muster in today's bioethical protocols. Granted, you know, 1961, 1962, things were different. And he said, are you calling me unethical? I said, no, I'm not going to judge you today by, what you did in 1962 or something. And he sort of stormed away and called me politically correct and, all this stuff. But it is really important, I think, to see how certainly in the last half century and in the last couple of decades, the entire protocol of interacting with patients and indigenous peoples from a standpoint of genomics has evolved. Which raises the question, of course, who is this research for? And as you remember from the Havasupai case not too long ago, it's always about getting the samples by talking about how important the medical benefits are going to be. But then there's a question of what actually are the medical benefits. And of course in the Havasupai case, they got the samples by talking about diabetes. And when people expected to see diabetes cured or at least studied, and instead found that they were studying schizophrenia and inbreeding, which led some of the Havasupai to think that they were being represented as inbred schizophrenics rather than diabetics. Of course, the research was being done for the benefit of the scientists, to keep the scientists busy, to get them to study the things that they were interested in. And it's nice to see that being reversed now. I can remember a story from about 1994. A distinguished anthropological geneticist was talking about his research on the genetics of alcoholism among the Navajo. And a Native American friend of mine asked him from the audience, "You don't really think that alcoholism in the Navajo is genetic, do you?" And he backed off very quickly and said, "Oh, no, only about 5% perhaps of alcoholism in the Navajo is genetic." And she said, "Well, then how can you justify taking money to study 5% of a problem and ignore 95% of a problem?" And his response was, "Well, I'm a geneticist. I study genetics," which I think really gets to the question of who is the research for. If the research is for the Navajo, then you should be studying the 95% of the problem. But if the research is for you as a scientist, okay, you're working on the 5%. And it's nice to see this sort of getting reversed now. It's interesting also, genetics is a lot better at talking about the future than at talking about the past. And the reason for that, of course, is that genetics is a highly probabilistic field, and the future is probabilistic. The past is history. And so nowadays, and that's why I think Rick's paper was very interesting, he talked about probabilities in the past. And one rarely hears that. One hears generally about the past, here's this migration, this is what happened. When in fact, a lot of those stories that get told, when you look at them 5 years later, 10 years later, they haven't held up. And to me, if you tell a story with the authority of science and it doesn't hold up, that strikes me as devaluing the authority that science is supposed to bring to the table. Let me say one last thing here, and that is a couple of weeks ago, Jerry Coyne, very distinguished Drosophila geneticist, was very critical on his blog, Why Evolution is True, which is intended for people still sitting on the fence. He was very critical of the use of the term identity, and actually wrote quite superciliously that identity is a term that's pretty nebulous and has no formal meaning in biological classification and even in biology. Now, I suppose if your frame of reference is fruit flies, all right, maybe they don't have identities. But I think this really does get to an important point about how different human genomics is from the study of the genetics of other organisms or the genomics of other organisms. This is biopolitical in ways that studying other organisms is not, and it's really important that we are acknowledging this, we are working within it. This is a multidisciplinary group of scholars talking about this and doing it from complementary perspectives. And I thank the organizers for inviting me in and sharing these thoughts with you.
Thank you all for joining us today. I think we're past time for a break, but we'll be continuing the conversations starting at 10:15 in Mile High 4A for the second half of this panel, and we hope you'll join us there. Thank you.